Hi again. So, having had an introduction to the methods we use in historical biogeography, we're now on part 3, which is a case study looking at the origins of insect diversity in the Western Indian Ocean, which is this region here, Madagascar and surrounding islands of the Mascarenes, Seychelles and Comores. And you see at first glance that they vary greatly in size, from Madagascar that's huge, through to some medium-sized and very tiny islands. They also vary in isolation. But importantly, they also vary greatly in geological origins and age. So some islands were formed by the fragmentation of the supercontinent Gondwana, that I'll come on to in a moment, and they're very old. These are the islands Madagascar and the Seychelles that I've marked here in green. And they have 60 to 90 million years of isolation as islands. While other island groups here are much younger and formed devoid of life. So I call these de novo origins. And this is either because they're volcanic, and this is the islands I've marked here in red, so the Mascarenes and the Comores, that vary in age from about half to 15 million years. Or they may have de novo origins because they're coral atolls or sandy keys. And these are the islands I've marked in blue here. And from a pure geological perspective, these islands may be as old as these other two groups of islands. Their origins are the same, but they've since been completely eroded and submerged. And submerged in recent sea level fluctuations. And therefore, from the perspective of biological colonization of organisms that we find there today, these islands have de novo origins and are very young, in the order of 10,000 to about 100,000 years only. So taking into account the parts we've just covered, so islands as model systems in part 1, and then the methods that we use in part 2, I want to ask the question, how important are the processes of dispersal and vivariance that I covered in part 2 in generating diversity? And secondly, how important have environmental changes been in generating the diversity that we see today, and on different timescales. So, including continental drift, for which the important events are really very, very old, volcanism, that's generally intermediate in age in terms of the important events for us, and then quaternary climatic changes that are relatively recent on an evolutionary timescale, and finally freak climatic events of the kind that we see today. So how has continental drift affected the Western Indian Ocean region? Let's look at a video. Of course, our understanding is continually being improved, but this is a recent summary. Okay, so here we have the supercontinents of Laurasia that you see here, and Gondwana that you see here. And you may not be able to see all the detail as I'm talking to you right now, but you've got the links there. You can go back and look at these videos slowly in your own time. They're kind of fun. What we're interested in for this session is what is effectively northeast Gondwana here. And you can see that nested in this region we have Africa, Madagascar that you see down here, and India. But do also keep your eye. You may be really struggling to see it here. In fact, I can barely see it, but it's a little white spot here. You'll see it more easily when you go through the video on your own. And this little white spot is the Seychelles microcontinent. And what we see here is eventually we're going to have the India-Madagascar block separating from Madagascar. There it goes. Then we have some back and forth movement between Madagascar and India. And then this is followed a bit later by separation of India from Madagascar, there it goes, and separation from the Seychelles microcontinent that you see I've got my cursor on it there. And now India goes zooming north to eventually crash into Asia. What did we see there? Well, a lot of movement and things going on, but importantly, 3 continental fragmentation events that have long been suggested to have influenced the native biota. So first of all, we see the separation of a block that includes Madagascar, Seychelles and India, and indeed also Antarctica, although we're not really interested in it from this exercise, it separates very early. We see the separation of this block from Africa around 130 million years ago. Secondly, we see the separation of the Seychelles-India block, this block here, from Madagascar at around 88 million years ago. And then finally, we see the separation of the India block from the Seychelles at 64 million years ago. And this is associated with what we call the Cretaceous-Paleogene boundary, which, as you may have heard, this was a period of Earth history in which a massive asteroid hit the Earth. But it was also a period in which the Reunion hotspot, that at that time was located under India, became active, giving rise to massive outpourings, massive volcanic outpourings that we call the Deccan flood basalts. So our question here is, can we kind of see the signature of this history in the regional entomofauna? That is to say, speciation events caused by Gondwanan vicariance. Well, to some degree, we can expect that geographic distributions of organisms reflect this history. And for many decades, distributions of taxa found exclusively on southern continents, as for example in this relatively recent publication from Earwigs Haplodiplates, this distribution was suggested to provide support for Gondwanan vicariance. Well, we can say here it's consistent with Gondwanan vicariance, this distribution, but it's not very strong support. It's better than nothing, perhaps. However, an uncomfortable result for the school of thought that proposed vicariance based on this kind of distributional data, which was popular many decades ago, this is actually a recent publication here, is that in the great majority of cases in which dated molecular phylogenies have been developed for such groups, so that is to say the methods that I covered in part 2, we find that divergence estimates post-date Gondwanan breakup by a really considerable margin. And this is true in insects as well as for other animals and also for plants. And therefore, most such relationships are now believed to be the consequence of long-distance dispersal aided by factors such as wind, cyclones, ocean currents, and also being carried on flying animals or on pieces of floating vegetation that can form natural rafts. And when I say carried by flying animals, I mean both on the feathers and on the outside of the animal, be it the feathers of the bird or the fur of a bat, or inside in the gut contents. Now, often when discussing this subject with students or the general public, I find that some take the same view that biologists did a century or more ago, that this sounds highly improbable, that a terrestrial organism like a flightless insect could cross thousands of kilometers of open ocean by these processes. But this, in fact, is viewing things from the perspective of our lifetime and the kind of processes that are familiar to us on our daily lifetime, in our daily lives and over our life experience. And we need to remember here that an event that happens only once every million years, so it seems almost impossible based on this lifetime perspective, is actually frequent on the timescale that we're considering here, of many millions of years. Of all the Western Indian Ocean insect cases for which there exists a dated phylogeny, for the moment, to my knowledge, there is only one for which Gondwanan vicariance is plausible. And this is the case of the Cascade beetles, in which Toussaint et al. Made divergence estimates based on 8 fossils using stem nodes for calibration. And as I described in part 2, this provides the most conservative estimate of minimum divergence times. And what Toussaint et al. Found is that confidence intervals of timing of divergence of the Madagascan and the Indian lineages overlaps with the breakup of these continents. So it overlaps with the Cretaceous-Paleogene boundary. So it's plausible that this divergence event was due to Gondwanan vicariance, but with some important provisos. So although there's only one species of Scoliopsis known, 7 species of Tritonus are known in the present day, of which only 2 here have been sampled. Should Tritonus prove to be non-monophyletic, that is to say, if we by sequencing another species, we found that that other species were to fall out here, for example, then it's conceivable that our true divergence that we're interested in of India-Madagascar would come out as younger. Similarly, the non-sampling of any unknown extinct lineages could conceivably have upset our interpretation in the same way. We don't know where those unknown extinct lineages could potentially fall out. So we need to keep these caveats in mind. Nonetheless, it remains a plausible case based on phylogeny and divergence estimates. And it also seems plausible given the ecology of the beetles concerned. So we can imagine that waterfalls are habitats that would have been buffered from the massive volcanism and the heat that would have occurred during the Deccan volcanism. Finally, note here that the low incidence of what I'm calling a Gondwanan vicariant signature in the living entomofauna of the Western Indian Ocean region seems typical across the tree of life and for other regions. So far, we only know of a handful of other such cases in a few groups, so amphibians, reptiles, plants, and possibly cichlid fish. It's not seen in other groups, so insects are no exception here. Okay, so if most living Indian Ocean insects owe their origins to dispersal rather than Gondwanan vicariance, where did they actually come from? Well, mostly Africa, we think, based on available dated molecular phylogenies. Nonetheless, data support dispersal from Asia for numerous lineages, and a good example is the millipede assassin bugs studied by Forthman and Weirach. And here we have a phylogeny that's been dated using fossil calibration, and ancestral areas have been reconstructed using the dispersal extinction cladogenesis model that I introduced back in part 2. And what these analyses support here is 2 colonizations of Madagascar that we see in red here and in the analysis here, and from Asia, or the Oriental region, that we see here marked in orange. So this is our phylogeny with our reconstructed distributions of ancestral lineages, and we see that here that we have, first of all, a speciation event associated with colonization of Madagascar in red from Asia around 48 million years ago, and that much later on, here in the phylogeny, we have again a colonization of the Madagascar region that we see here from Asia occurring later around 28 million years ago. And this was also followed by one colonization of Madagascar from Africa that we see occurring here, this event here, at around 20 million years ago. So what we see here is a significant proportion of lineages arriving in the Madagascar region from Asia. Well, that lineages dispersed to the Madagascar region from Asia can seem surprising, given this huge expanse of open ocean to be crossed from India to the Madagascar region when compared to the much shorter distance that we see from Africa. But it perhaps seems less so when we consider the direction of prevailing winds and currents, which go predominantly, though not exclusively, from the Asia region towards Madagascar and therefore towards our Western Indian Ocean region. It can also seem less surprising when we consider the effects of quaternary climatic changes. So at repeated intervals during the Quaternary, which is the last 2.6 million years, sea levels were much lower due to climatic fluctuations. This lower sea level would have exposed numerous islands. So some islands we know today would have been very much larger in size. So we see this map looks very different. Back at times of low sea level, we see that the granitic Seychelles was huge compared with in the present day. Same for the Maldives and the Chagos, but we also see numerous islands that don't exist at all in the present day. So Sayed Amalha, Nazareth, and St. Brandon. And together these islands may have served as stepping stones, reducing the distance of open ocean to be crossed from Asia towards the Madagascar region. So these stepping stone islands existed not only during quaternary low sea levels that I mark here in blue, their origins, which I mark here with red lines, are age progressive, relating to plate movement over the Reunion hotspot. First of all, the Indian plate, and then later the African plate. And this began with the Deccan flood basalts at the Cretaceous-Paleogene boundary, and it's been age progressive right through to the present, with the Reunion hotspot currently, of course, under the volcanically active island of La Réunion, which you will have heard on the news just recently, has erupted just this year. Note that this case here for the Indian Ocean is very much like the Hawaiian case that we covered in part 1. So here I've marked all the published estimates of insect arrival times that I know of to date, based on molecular data, molecular phylogenetic data, and from all continental sources. And we can see that cascade beetles really are the exception here. All other cases are much more recent and therefore must relate to dispersal. It's the only explanation that remains for us. Okay, next we will come down in geographic scale and ask, once a lineage has arrived from a continental source, how is diversity generated within the Western Indian Ocean region? And this is an example from curculonid beetles, weevils, c'est-à-dire charançons en français, in which we've used an ancestral area reconstruction method that I introduced in part 2, so the dispersal, extinction, cladogenesis model, to reconstruct the biogeographic history of divergences in this group. And so here once again we have our ancestral area reconstruction plotted on the phylogeny. Once again, each pie chart here represents our reconstructed distribution of an ancestral form in the phylogeny. So you see here the code for the different geographic regions concerned. And so what we can infer is that speciation here has occurred mostly by inter-island colonization. That is to say, colonization of one island from another island, and that there's sometimes been multiple such inter-island colonizations. So in effect, often back colonization of the original island. So if we take, for example, this clade here, we can infer that the ancestral weevil was on Mauritius here. that this divergence event here was associated with the colonization of Reunion, which is in purple. And at a later point in time, still we have the colonization of the back colonization of Mauritius from Reunion. At this point here, in which we see that it's rather small here, but you'll see it if you zoom in on the slide, that one of the two descendant species is found on Mauritius today. As mentioned back in part 2, ancestral area reconstruction, in general as an approach, provides our best estimate of where a lineage occurred based on assumptions. But it remains an estimate. It is only as good as those assumptions hold true. And so, also as I mentioned in part 2, a useful alternative is to simply consider by hand what the alternative biogeographic histories are based on the phylogeny and the geographic distributions. And this is an example of where we've done so for a group of crickets, Ornibius, in which we find two scenarios are highly plausible because they require a minimal number of steps. So scenario A and B. So first, some generalizations. We infer that speciation under either scenario, A or B, has occurred mostly by inter-island colonization. So that is to say, on this graphic here, these events here, where a divergence event goes from one island to another. That's the predominant story in either scenario. Under scenario A, that we have here, we have ecological shifts from lineages that are associated with dicots, that I've marked here in brown, to lineages being associated with monocots, and specifically the monocot pandanus. I can suggest here that this shift in ecological association has permitted islands to be colonized multiple times under scenario A. So here we have a lineage on Rodrigues that's colonized Mauritius. It's become associated with a monocot, and I would suggest that this association here has permitted this lineage to then back colonize Rodrigues, and if you like, to live together with the original dicot-associated lineage on Rodrigues. And although we see some within-island divergence in this first scenario A, and that's right here at the end, this divergence here between lineage 5 and 6, which are not full species, but they've diverged a little bit they've diverged in song, not in any other features. Although we see this one case here on Reunion, we otherwise do not see any within-island speciation, full speciation events occurring under scenario A. By contrast, under scenario B, two within-island speciation, full speciation events are inferred. One here on Rodrigues, and another one much later on Mauritius. And these we can infer were likely associated with shifts in ecological association. So once again, a shift here from dicot association to monocot, and then later a switch back to dicot association. So of these two, I slightly favor the second scenario, but based on the data we have, these both seem highly plausible. And what's worth noting is that some within-island divergence has occurred in either scenario, A or B. This case here of this divergence, small genetic divergence and divergence in song. And this seems to be common in many clades, many Indian Ocean insect clades, that they form monophyletic groups confined to particular islands. So this leads to the question, what actually drives speciation within islands? I'm taking here an example from Mauritius, which has its volcanic origins dating back 7 to 8 million years. And there's then been two further episodes of renewed volcanic activity. one occurring at around 2 to 3.5 million years ago, and a second one that's more recent, occurring within the last million years. And the result of these periods of volcanic reactivation is that you effectively have in the present day a matrix on Mauritius, that is to say, blocks of ancient rock surrounded by a matrix of much younger rock. So effectively we have kind of islands within islands. And the mountain of Le Pouce is a good example of one such geological island, a block of old rock surrounded by a matrix of younger rock. And what's interesting is that many cryptic insect species seem to be endemic to this mountain, both the mountain top and the surrounding forest. And this is based on work in progress. By this, what I mean is that there's genetic lineages that are highly divergent from lineages found elsewhere that are only found on this mountain or on the high forested slopes of this mountain. These lineages were not previously recognized as species due to a lack of morphological variation. So there's a growing recognition across many geographic regions with genetic data of cryptic species. And once again, I emphasize this is work in progress, but our provisional results suggest that we may have here 3 cryptic orthopteran species and 2 cryptic cratopus species, 2 weevil species. And therefore, we may perhaps be able to talk about Le Pouce as a hotspot of cryptic species diversity. Well, how do we explain this? Well, Le Pouce Mountain certainly represents a refuge of good habitat, relatively pristine forest, surrounded by human-modified habitats. So you see here, as you see here, there's a lot of sugarcane, there's a lot of urban areas. So perhaps you can argue this concentration of locally endemic and cryptic species can be explained by habitat distribution alone. Well, this is a possible explanation, but at the moment to us it seems weak. First of all, because there exist other patches of good forest where no such local endemics occur, and also because when we look at the sister to these cryptic species here found on Le Pouce, they often occur in highly human-modified habitats, as is the case here for cratopus, in which this sister lineage here occurs on offshore islands that have had their habitat highly modified. Rather, we think that the history of geological reactivation at the moment seems a better explanation, that we effectively once had a widespread species across Mauritius, and that there was then a lava flow that came across here that we know about from the geological data, and that this would have fragmented the range of these widespread species, leading to speciation by vicariance, that this lava flow would have been unfavorable habitat to the insect. Of course, so yes, this remains a work in progress, it's a hypothesis. Of course, coming back to the general case of insects on Western Indian Ocean islands, other mechanisms may also have contributed to speciation within islands. And in our ornibius cricket case that I covered earlier, ecology also seems likely to have been important. So that may have been a major factor, or the two may have worked together. Another general thing to note is that insects contrast with other groups in their ability to speciate at very small scales within islands. For example, this never occurs in birds, which are my main study group, on Western Indian Ocean islands that are smaller than Madagascar. And this finding for the Western Indian Ocean birds is actually consistent with data from birds worldwide, that speciation very rarely occurs within islands that are smaller than 10,000 square kilometers. So what have we learnt in this session about origins of Western Indian Ocean insect diversity? Well, first of all, how important are dispersal and vicariance in generating diversity? Well, as a quick generalization, the data suggests that dispersal has been very much more important than vicariance. But it's important to qualify this with some detail. So, speciation through dispersal is the dominant source of diversity in all insect groups. It seems responsible for at least half of the speciation events that we've documented. And it's occurred through both colonization of archipelagos and inter-island dispersal events. These latter inter-island colonizations have often been repeated. So we've had multiple colonizations of the same island within a single clade. And these inter-island colonizations may have been facilitated by ecological shifts. Vicariant speciation is possible through two different kinds of geological processes. First of all, continental breakup is a nice idea, but at most it seems to explain a very small minority of living insect diversity. We've only found one plausible case so far. Concerning volcanic reactivation, volcanic activity, its importance seems to vary depending on dispersal abilities. So it may explain a moderate proportion of diversity, up to half of the speciation events in poor dispersers, such as the Nepheliophila that we covered in our Lapus slide, but it seems to explain a minimal or zero contribution to diversity within groups of higher dispersal capacity, such as the ornithibius crickets that we covered in an earlier slide. Second, what has been the likely importance of the following environmental changes in generating the diversity we see today? Well, as just discussed, although continental drift could generate diversity by vicariance, this only very rarely seems a plausible explanation for the data. So I've marked its importance as low. Regarding volcanism, you'll see that I've marked its importance as medium. It has certainly been an important contributor to the formation of new islands, and the colonization of such new islands has given rise to many species. At a different scale, within island vicariance may likely have occurred due to volcanism, and this in turn has been an important contributor to the diversity we observe today. Regarding quaternary climatic changes, we learned that, first of all, we learned that such changes in climate led to changes in sea level that in turn led to changes in island area. Important changes in island area, you'll see nonetheless that I've marked this importance here as low, with a qualification. So such changes in island area were likely critical in allowing waves of immigration to the Western Indian Ocean, and particularly from Asia. However, most lineages we know from the data have been through at least 30 such cycles without speciating. And so at least in the Western Indian Ocean region, there's not been a species pump driven by such climatic changes and changes in island area, as has been suggested for other regions of the world, that such climatic changes drove diversification. Regarding freak climatic events, you'll see that I've marked their importance as putatively high. We can expect them to have been important insofar that data support chance, that is to say waif dispersal events, as the dominant factor in driving inter-island colonization, which in turn has been the dominant factor in driving divergence. It's the only explanation available for many such colonizations and associated divergences. However, the word putative here, I've marked here, is important. Of course, we've not tested to what extent stochasticity in dispersal and survival of insects alone can explain this, and to what extent freak events like major storms and cyclones known to pick up insects can have facilitated dispersal. Their importance cannot readily be tested with genetic data. Rather, if anything, I guess that advances in technology for tagging insects, once they become effective in the future for many insects, might one day be informative in this regard. So, wrapping up, what can we draw from our case study about origins of diversity in a changing world? Well, first of all, some prudence is definitely called for. Much insect diversity remains to be documented. The work ahead for taxonomists remains enormous. And of that insect diversity which is documented, genetic surveying, that is to say, the construction of molecular phylogenies, as we've been dealing with here in part 3, they're far from being complete. Much work remains even for the documented lineages. Furthermore, some hypotheses merit further testing beyond the capacities of genetic data, such as the processes of wave dispersal that I just discussed in the previous slide. Nonetheless, it's really no exaggeration to say that adding molecular data for model systems such as island insects has really revolutionized our understanding of the processes generating diversity at a regional scale. In most cases, just 20 to 30 years ago, we just simply have had no detailed information with which to respond to the questions that we just addressed. So in coming to an end, I hope I've left you with an impression of how island insects are kind of cool systems for understanding diversity in general.